Okay, welcome everyone. Thank you for coming. This is our third, I guess, distinguished lecture in the series this year. So, yeah, well, let's, why don't we get started? I will have Patrick Park introduce our speaker for today.
Hello, everybody. I have the pleasure of introducing Professor Carter Butts today. Carter received his PhD here from Carnegie Mellon in the Social and Decision Sciences Department, doing a mathematical and computational social science thesis. He went on to win the Freeman Award from the International Network for Social Network Analysis, given to a distinguished young scholar in the field of social networks. And part of it was for his groundbreaking work dealing with bias estimation. He helped develop some of the original network science algorithms in R, and the community recognized his contributions through the William D. Richards Software Award in 2019, I believe. Today, he's considered one of the top mathematical and computational sociologists in the world. His interests are broad and highly interdisciplinary, spanning mathematical sociology, economic sociology, social and biophysical networks, computational, Bayesian and applied statistics, simulation and computational methods, molecular modeling and analysis, human judgment and decision-making, and disaster research. He's currently a Chancellor's professor at UC Irvine in the departments of sociology, statistics, electrical engineering, and computer science. He's also a member of the Institute for Mathematical and Behavioral Sciences and an elected fellow to the American Association for the Advancement of Science. A fun fact about Carter is that his first publication was in the first international conference on the World Wide Web, also known as WWW1 or the Woodstock of the Web, back in 1994 in Geneva, Switzerland. He was an undergraduate first author of the paper.
It's alphabetical though.
Oh. I was going to say, yet he claims his quote dubious contribution was writing an email to the WAIS gateway. So without further ado, I turn the floor to Professor Carter Butts.
Thank you. I appreciate the introduction. You're supposed to, and this is especially for the students and postdocs in the room, you need this bumper sticker for like, what do I do? When people ask you, what do you do? I don't have that anymore. I can't explain it anymore. So that's, it's a good explanation. I can't. I work on a lot of fun stuff. So anyway, it's wonderful to be back. Thanks for the invitation. Thanks to you for coming out today. It's been a long time since I was on this campus, but it always feels like home. It's an exciting place where people are always doing cool stuff in a way that elsewhere not everyone is, which is something that some of you will grow to appreciate when you're not here. But anyway, that's life for you. So enough of my easering at you. Let me tell you what I'm going to talk about. So I'm going to talk today about some work that I and some colleagues have been doing over the years, and some more recent, looking at the problem of modeling sets of networks statistically. And I'm going to approach this in a particular way. There's lots of ways people think about this problem these days. I'm going to use a very traditional Carnegie Mellon way of doing it, or at least the way that it originated here, which is I'm going to do this using exponential family models. And these originally came out of work that Holland and Lionheart did, and of course, contributions by the late, great Steve Feinberg, among others, for modeling networks that have dependence among edges. So the thing that makes networks hard to model from a statistical standpoint is that the ties in one part of the network are affected by the ties in other parts of the network, right? If you and I have friends in common, we are more likely to be friends than if we didn't. All other things held constant, and that makes statistics on graphs fun or hard. Well, let's stay fun, okay? And so there's a lot of ways to think about that, but what we tend to do these days is use exponential family models. So we write them down in these forms that look like this. So this is an exponential family random graph model, an ERGM form. Some of you are very familiar with this equation. Some of you are like, what is that? I will give you just the bumper sticker idea, and I'll show you an example in a minute. So the idea is that we have some random graph y drawn from some support script y. I'm going to assume today it's finite, although we can relax that. If it's finite without loss of generality, we can actually write it down in this way. And so what are the elements of this? This thing T here is a vector of sufficient statistics. If you like, think of them as features of the graph, possibly in association with covariates, X. They could be counts of triangles, although I wouldn't recommend that one. They could be correlations of degrees with some attribute, whatever they may be. We're going to, in a sense, score every graph by looking at these statistics and weighting them by these parameters, theta. We want graphs that have higher scores to have higher probabilities, so we exponentiate them to make them non-negative. The then has to be normalized to make it a probability, so we just add up the same thing over the support. And then we have this funny thing H here, which is a reference measure, and that controls what happens to the model when theta goes to 0. And there's actually kind of deep interpretation of H, but you can also, for the moment, just think about it as setting the baseline behavior of the model. In a lot of the work, you see H is just taken to be a constant. That's the same as using the counting measure on Y. But in more recent work, we've started to elaborate this. And so, for instance, when we want to keep the mean degree constant as n changes, the size of the graph changes, we use a reference measure that looks like that. So modeling of this kind is a big industry these days and is increasingly mature. I wouldn't say it's done by any stretch, but we do have tools out there that if you want to do maximum likelihood inference first data, we have ways to do that. Geyer, Thompson, Humble schemes, Robbins, Monroe. We have Bayesian methods, approximate techniques and so forth. And so we can do it. It's still hard. There are a lot of really cool computational challenges. That means that people like me can write papers involving this normalizing factor. So it's still an active area. But these days, if you have kind of reasonable models, reasonable data, you can usually fit stuff and it kind of works. So let me show you an example to fix ideas. If you want to see one of these in actions, and I'm going to use again a Carnegie Mellon example. And indeed, the perpetrator of this is in the room with the data. So this data is collected by our own David Crackart here some years ago in a high-tech firm. So each of these nodes here is a manager in the firm. And I have colored the nodes by the department they're in. And there's a tie between the two if they consider each other to be personal friends. I'm using even David's favorite way of talking about personal friends, which is they both have to agree that they are personal friends. Okay, so this is Crackart safe or endorsed. Okay, you may not endorse the rest of it, but at least the data. You can't blame me for that. All right, so we think about what might drive the structure of that friendship network. Well, we can think of lots of things that could go on, but one basic mechanism that we know it's often important is homophily. We might think, okay, people in the same department, they interact with each other a lot, they have things in common. These may make them more likely to be friends or stay friends possibly. And if we eyeball this, we use the interocular percussion test, as we like to say. Well, it does look like there are more ties maybe inside groups, but I don't know. There's some that aren't there, so hard to say, but maybe. Of course, we also have triadic closure, right? Friends of friends tend to become friends. And if we look at this and we say, well, there are a lot of triangles, we suggest that, well, there look like there are a fair number of triangles. Of course, are there a lot more triangles? And are there more triangles than you'd expect from homophily? Well, interocular percussionists not so good at that. That's why we have statistics. Okay, and so we can fit a model to this graph using our ERGM framework to look at things like that. And the way we specify it is we have statistics that represent those mechanisms. So the edge term represents an overall propensity to form ties. We have something called node match, which represents a propensity to form ties within groups. And then we have this complicated thing called GWESP that represents a kind of triadic closure bias, but in a sort of safe and friendly way. And we can fit the model to data. We get MLEs, we get standard errors, we can do our usual beautiful statistical things. And indeed, we find, yes, net of everything else, there is a tendency toward homophily. It's pretty strong. There is a tendency net of that to triadic closure and so forth. And because we have a generative model, we can also simulate realizations from the graph. So we can actually say, okay, given that model, what networks would I expect to have seen? And are those networks similar to the one we saw, right? So we can do model adequacy checking. We can say, all right, is this model reasonable enough? It's a very, very simplistic model. We know that, but can it reproduce the types of properties that the graph has? So we can look at, say, the degree distribution, guide-wise shared partners, edge-wise shared partners, the geodesic distances, and the actual data is here in black. These intervals represent simulation. And yeah, the model, as simple as it is, does a good job of recapitulating. Okay, so that's a very, very simple case, of course. That's a toy example, but it shows you some of the kinds of things that we can do and we do routinely with these models. All right, fair enough. That's one graph, one graph. That's for wimps. Let's make it harder. Let's think about what happens when we got multiple networks. And there are a lot of problems, many of which I will not talk about today, that involve multiple networks, right? So the most obvious case is if I've got a population of graphs, I observe some sample from that population, and I want to infer something about or predict something about the population of graphs. Maybe I have a bunch of groups or organizations or systems with network structure. There are other cases too. Maybe I observe a bunch of graphs that are all noisy measurements of a single underlying graph, and I want to figure out what that graph is. So a measurement problem, that's a multiple graph problem. What if I have a time series of networks? Okay, that too can be a multiple network problem. We can have lots of different ways of like EGOT-nets. There are lots of things that lead to this. So there are a whole bunch of different things that involve simultaneous analysis of multiple networks. Today I'm going to be focusing on this type of problem, right, where we have a sample of networks from some kind of population, and we're interested in drawing some conclusions about it. Now, when we think about that, I've got a bunch of data sets. In essence, if you think of each network as a data set, and I want to learn something about them collectively, if you want to think about how to solve that problem, that's a general problem that in statistics we think about a lot. And if you come from a statistical background, you probably think about these things as living, the solution to these problems is living along an axis. The colors didn't come out so well. One of these is more blue and the other is more red, but you can't see in this projection, but that's okay, it doesn't matter. At one end of the spectrum, what we do, well, we have independent estimation, right? I can take every network and fit a model to it, and then either be done and say, that's great, now I have a bunch of models, or maybe I take the parameters of those models and I do a second stage analysis of the parameters. There's nothing wrong with that. And in fact, that's the most non-parametric thing you can possibly do, right? Let every data set speak for itself. Of course, there are problems with that. You're not pulling any information across cases. If some of these are hard to fit, well, you're SOL, but you can do it. Now, at the other extreme, we can do pooled estimation. We can say we're going to have one model to rule them all, treat them all as approximated by the same generating process and fit one model. Maximum data brought to bear on those parameters, but of course, homogeneity assumptions, right? Not always a good idea. And in the middle, well, there's all the other stuff, partial pooling. Often we think of this in the context of hierarchical models, right? Where we have some sharing between, but not 100%, right? And of course, in this, all sorts of methods live, right? And that we can think of a lot of things. So today I'm going to talk about a few different examples in here. I'm going to start over on this side, and I'm going to talk about some things that work toward the middle. I'm not going to talk about this. It's sort of obvious. And the first time is I'm going to go all the way over here to pooled estimation. And you might think, well, that's kind of boring. We already know how to do that. But there is a challenge that isn't so obvious. And that's how to do scalable pooled estimation. So that's what I'm going to be focused on. And the example I'm going to use is a bit different from the friendship network. I'm going to talk about some other work that we've been doing recently with Doug Tobias's group at Irvine on aggregation of a protein called WD crystalline. So WD crystalline is a protein that you know and love, even if you don't know that you know and love it, because it's in your eye lens, and without it, you couldn't see my talk. Well, okay, maybe you're now having second guess thoughts about your gamma D crystalline, but it's a structural protein in the human eye lens. It's mostly expressed toward the middle of the lens, and it has a very high refractive index, and it's part of the thing that determines the refractive index of your eye, without which you would not see. Now, part of it is, of course, water. That also contributes to the refractive index of your lens, but the crystallines are very critical. These are really interesting proteins for a lot of reasons. One is that you're endowed with a certain number of them in early development, and you don't get any more ever again. They cannot be repaired. They cannot be replaced. So if they go bad, you go blind. And in particular, they stick together, they scatter light, and you get cataracts. So that's where cataracts come from, is that mechanism. It's the world's leading cause of blindness. And indeed, the aggregates they make in this sort of complicated form, some examples of electron microscopy of these things, but bottom line is they're bad. Now, what's interesting is that these things manage to last for decades, an incredibly high concentration, about 500 grams per liter in the eye lens, which is an enormously high protein concentration without ever kind of sticking together, except when things go bad, in which case they do. And so how does that work? Well, that's something people want to understand in order to understand the cataract mechanism as well as protein interactions in general. This is a hard thing to study, especially computationally, because the workhorse technique that we use for modeling proteins, atomistic MD, molecular dynamics, is out of scale here, right? If I take one small protein, just something like A beta, which is a lot smaller than this, and I run it for a couple of weeks on a GPU machine, simulating it, I can get about a microsecond of simulation time. Okay, your eye lens, right? You hope that it's going to last you for many, many decades of your life, right? Before aggregating. So that's a lot slower process. And you're talking about many, many, many proteins, not just one. So it's way out of reach. So what do? Well, people, of course, work on this. And one of the things that my collaborators have developed is a particular kind of atomistic Monte Carlo scheme that does allow us to get snapshots of the aggregates that these things form for hundreds of proteins. At sort of realistic concentrations that it can't go up to the very highest, but they're still useful. The problem is even these things require like months of supercomputer time to run. So a question is, can we come up with a simpler, so these things are hundreds of parameters, simpler models that are much faster that we can simulate in a reasonable way that would let us reproduce some of the processes of these aggregates. And what we're going to do is we're going to think about doing that using ERGMs by thinking about the aggregate as a network, where every node is a protein, and there's a tie between two proteins if they're stuck together in the aggregate. And so what we're going to do is, as we're using here as data, are the simulations that came out of this atomistic high-quality method. And we're going to see how to do that. So what are these things like? Let me show you a bit about this. And I want to highlight my student here, Liz Diesner. It's done a lot of the work on this project. All too soon, she will be on the postdoc market. Sorry, not looking for a postdoc. We got people here we hook you up with. All right, so what is this thing like? So if you're not a protein enthusiast, this is what the monomers look like. So here in blue is the wild type. That's the friendly version. And the one in red is the not so friendly version. And even if you're not a structural biologist, you can kind of see this thing is kind of wonky up here. So what's happened is we've got a tryptophan to arginine substitution in the N-terminal domain. It kind of disorders it. It exposes hydrophobic, oily bits of the protein, and it gets sticky and it sticks to stuff. And that leads to differences in the aggregate size distributions. Down here at the bottom, I have shown the distribution of aggregate sizes, a log scale. So for both wild type and the cataract-prone mutant, these fall off is about 1/n squared for a fairly large range, but wild type starts to peak out at about 100. Okay, so it doesn't make really big protein aggregates, but of course the W42R keeps growing and makes rather larger aggregates. What do they look like? Well, these are both representative graph draws from our distribution. So this is what they kind of look like. So each one of these nodes is a protein, and they're tied if they're stuck together. And we can see they make these kind of loose, relatively sparse aggregates, but they do have some local triangulation. And if we squint, we might say, well, maybe this is a little more extended. Again, squinting isn't good enough, right? We want to actually model. So what have we done? Well, what we've done is we modeled these, again, using the ERGM framework, and we can do this using the same kinds of tricks we used with the friendship network. Slightly different statistics, although many of the ones we use are, in fact, ones that we use for social networks as well. So we have things related to, of course, edge formation. We have various closure-related terms, and we've added some terms that are related to the distribution of component sizes. And what those things can be thought of physically doing is expressing differential tendencies for big aggregates to hook on to other big aggregates or not, right? And so some things they tend to fold up, make fairly kind of enclosed sort of spherical things, and they can't hook to their neighbors as well. Other things can be more free and easy, and they can grab on. There's some corrections that we have to do that I'm not going to talk about unless you ask me. And if you ask me, I'll tell you more than you want to know for like bond energy thing. There's some really fun stuff, but we're going to fit a model in, but we're basically using the same tricks that we used in that friendship network. One of the things we are going to do that's a little different here is that because our goal is to get the aggregate size distribution right, when we do model selection, we do it instead of by optimizing AIC or BIC, we look at the L2 norm of the log relative distribution for the observed versus simulated component size distribution as a way of picking. So this is the case where we have a concrete prediction target. We're going to optimize the model to reproduce that target, but otherwise it's the usual deal. So all of this is straightforward, right? Let's just fit some ERGMs and go. Well, there's one problem. We have 15,000 networks, actually, each for the two different cases. That's a lot of networks. Now, how do we do pooling? I haven't told you how we do pooling. Here's how we usually do pooling. We take each of the networks, we stick them all together in one huge meta network. We put some block constraints on it to prevent ties between the blocks, and we fit one giant MC-MC chain to the entire thing. And that's not bad if you have small networks. The problem here is we got 375 nodes per network and 15,000 networks, and that makes too big. Okay, so we can't fit that. It's not going to work. So we need a trick. And so what we're going to do is use one weird trick which is in a paper that just came out a few months ago that my now former student is now in industry, Fan Yin, and I wrote, where we exploit a really simple property of exponential families. You can look at the paper when you want to find out how simple it is. If you don't believe me, it's embarrassingly simple, but somehow no one had gotten around to using it in this setting. And what it lets us do is that in certain cases, of which this is one, we can have a whole bunch of input networks, and to fit a pooled model to them, instead of fitting a model to this giant meta network, what we do is we fit one network where we fit to target mean statistics, sufficient statistics, that are just the averages of our original. So it's something so stupid, you think there's no way it can be this easy, but it is. Okay, so we have one representative network we fit to with target sets that are just the mean of our other statistics. And that means the cost of 15,000 networks is, other than a little pre-processing time, the same as fitting one. Now, of course, we got to correct standard errors because we didn't have one network at 15,000. That turns out to be a linear correction. Nothing in life is ever this easy, okay? But it is, and it turns out it works. In fact, you can also use this trick to do map estimation for free, but I'm not going to talk about that other than highlight. Go read the paper, you can see. But anyway, so this allows us to take this 15,000 network case and fit it directly. And that's exactly what we do. When we do, what do? We get parameters, okay? And one of the fun things, which again, I'm not going to go into detail unless we want to hear more later about it, is that we can express our parameters in the usual ERGM way, but we can also actually express them in terms of a Hamiltonian governing the system, because we have actual temperatures here and whatnot. So I can give you actual energies, kcal per mole contributions, which is great when you want to communicate with physical chemists in their language. So we can actually talk about this in ways that make physical sense. But basically, what do we find qualitatively? Not surprisingly, W42R is stickier. It does, net of everything else, have a greater propensity to form ties. We see slightly different closure behavior. In essence, the W42R closure profile is a little flatter. So there are fewer diminishing returns for if I give you one other shared partner, another shared partner, another shared partner, adding to closure. The thing that matters the most, though, is this funny little thing down here that you probably think, well, this doesn't matter. Look how small these numbers are. And this has to do with the propensity of big components to stick to each other. And there's a little bit more of a tendency for big components to not stick to other big components for wild type versus W42R. And this is actually the key thing that matters. W42R makes these kind of more extended components, and the big ones will hook on to other big ones. And that lets it get bigger and bigger much more readily than wild type. And so this is part of what seems to be governing that, that growth. Now, does it work? Yes. Okay. Does it reproduce, at least does it reproduce the observed distributions? It does pretty well. It's a very simple model, only a few parameters, but we can do a pretty good job. Here's our observed versus modeled size distributions for the aggregates, W42R wild type. And then these are local properties, degree distributions, ESPs, which we really kind of nail. So again, a very, very simple model, much simpler to put into it, but it can recapitulate this kind of complex structure. So that's one example, right, of a multi-network analysis. That's an easy one in a way because we know this has a single generating process that we want to characterize. Well, what if things are more difficult, right? Let's go back to our range. Okay, what if I want to back off from pooled estimation, right? There are a lot of cases where I think some things are going to vary between networks. And if I want to pool a lot, but allow some things to vary, a natural place to go is mixed models, right? And there's work on that, including she's unfortunately not able to be here, but Nikki's group has been working on mixed models. She's done a lot of stuff with P2 models, for instance, where you have some effects in the model that have a stochastic structure and then other things are fixed. So that's sort of one way to grow. Now, when you actually do it like her way, she's got a macho way to do it, right? Where you have a whole stochastic process on top of the parameters and all that's really cool. But we might be lazy and say, is there a way I can do this less macho? I love one weird trick. I'm going to give you a trick for doing this, at least in some cases, without having to do that. But again, the same idea, basically. We're going to treat some effects in a hierarchical way and other effects pooled. And the way we're going to do this is by going back to basics and thinking about, at first blush, something that seems much simpler. And that's thinking about really, really simple, taking the simplest model, really, that you could have for a network and making it a little more complicated. So the simplest model you probably have for a network is a Bernoulli graph. So think about a network where every tie is an independent Bernoulli trial with some probability delta. And if you want to make that a little more complicated, one of the things that you could do is say, what if I let that delta, instead of being some fixed value that I give you, what if I draw that from some distribution F? And that's not per edge, that's just for the whole graph. So I have a graph where I'm going to generate the density from a distribution and then generate a Bernoulli graph. And this is actually an old idea. Frank talked about it in the 80s. Didn't do too much with it, but mentioned this idea. And so it's a very natural thing to do. And this actually can be a really natural starting point for hierarchical modeling because we can start by working with models that are these mixtures, and then we can put extra structure into them. So how can we do that? Well, let's start at the beginning. So with the Bernoulli graphs, we can think of these as density mixtures, right? So we're going to draw our expected density from some distribution F, and then draw a Bernoulli graph with it. A natural choice here, we could pick anything for this, but a natural choice is a beta distribution. And if we do that, it turns out we get a family of graphs that we can write down in closed form that I'm going to call the beta Bernoulli graphs, in analogy with the beta binomials, if you're familiar with that. And in fact, the counts of edges are beta binomial, but it's going to be a graph. It's just mixing across densities. We can do the same thing if we are in a directed case and we want to mix across reciprocity levels. So we can take, so every dyad is going to, and that's a graph, is going to be mutually asymmetric or null. So we can think about generating a network where every dyad has a chance of being mutual, two ways, a chance of being asymmetric or a chance of being null. And again, I can generate those probabilities from some distribution. A Dirichlet is a natural choice. And then I can generate a categorical graph, right, which has those distributions. And that we're going to call the Dirichlet categoricals. And these distributions are neat because unlike most things, you can write them down in closed form. I can write down, now they're a little ugly. Okay, I mean, maybe you love the gamma function. If you love the gamma function, it's beautiful to you. But anyway, but it's there. I can write it down. And I can even write it down in a form that looks a lot like a curved ERGM. Okay, so where I've got these terms that involve log gammas of the edge counts and the null counts plus parameters. This is just written for multiple networks. And we can make that look even like a canonical form. We could expand these curved things and get canonical form. Now our statistics are the powers of the edges and the nulls, and our parameters involve derivatives of the digamma function. So we can do that. Now, if you're an exponential family purist, you're frowning inside or outside because you're like, that's an infinite number of parameters, but don't worry, I'll make that problem go away. We'll make it all go away. We can do that for the Dirichlet categoricals. Again, we get a wall of math, but what's going on, we can write it down in terms of, again, powers of mutuals, asymmetrics, nulls, and this is just a correction for the fact that there are two different directions that an asymmetric can go. So it's relatively straightforward. Okay, now how does that help us with ERGMs? Okay, these are still just mixtures of boring baseline graphs. Well, now that I've written in something that looks like an exponential family form, what I could do is I could take the baseline, this is our beta binomial case and our beta Bernoulli case, and I can just add more ERGM terms to it. I just stick them in here, right? These would be things like West and all that stuff. Why not? I can write it down. I can do that if I want to. And what's that going to be? This is going to be analogous to a mixed model on the edge parameter in essence, right? Because it starts with, instead of starting with a Bernoulli graph, it's going to start with something like a mixture of Bernoulli graphs, but now we're going to have these other effects in it. And I can do the same thing for the Dirichlet categoricals as well. And again, I have this Dirichlet categorical ERGM where it's like I'm mixing over reciprocity and density. And I'm allowing that to vary by graph and then having the other effects constant. Now, of course, I have this problem of I got these infinite terms and what do I do with that? It's displeasing, but we can fix that at least approximately. We use a trick involving the properties of the gamma function. And it turns out we can write those curved things to a very high degree of precision in practice with just two terms. So we wind up with an offset that's just the log gamma function of either the edge count or the null count. And then the statistics that we wind up in the model are just logs of the edge and null count. So we wind up with a really simple ERGM family where we have two offsets and then two sufficient statistics here. And although we can't, these are edges and nulls in the same model, we can't identify them with one graph, but if you give me two or more, I can, which makes sense because it's a mixture, right? We do the same trick with the DC family. And again, it's the same idea. We again have logs of mutual asymmetric and null counts being our statistics. Great thing about doing it this way is this is something that you can plug into existing software. It would take some work, but you can use, for instance, the StatNet toolkit, and you can write down change scores for these things, and you can put them in and you can fit ERGMs with it without having to introduce. So this is the advantage of doing it in the less macho way, is that instead of having to build a whole additional apparatus, we can use all the standard model fitting techniques we use and simulation techniques and computational techniques by changing instead changing the term. So what we've done is in essence collapse that mixture, and then we're able to work directly with that. Okay, so let's see an example of this thing in action. I'm going to show one of my favorites. This is a great data set collected by Zablocki, published in the 80s. It was collected in the 70s. Things were different back then. Okay. Despite the hair, I have nothing in common with these people. Okay. I'm a tech, not a hippie. Okay. They don't wear ties for one thing. So, okay. But so what Zablocki did is they went to a bunch of urban communes in the US. There were, well, they still exist actually. There were those things. And they interviewed people and they asked them all kinds of crazy questions. This data set is wild. Okay. I don't know any other data set where they ask people to say, who in your group do you consider holy? Okay. Or sexy. And there's a great, John Levy Martin has a great paper called Is Power Sexy? Okay. This is science. Okay. We have answers to these questions. And we can do this with this data set. What I'm going to do is I'm going to look at perceived power. So one of the things they ask people is does one of you in your relationship, and they ask this for each pair of people, do they have power? Do you have power over them? Do they have power over you? So what we're going to do is we're going to look at each commune, and for people I, J in it, if I or J says that I has power over J in the relationship, we're going to say there's a power in that. Okay, and there's a tie here from I to J. So this is a power relation, perceived power relation. This is something that, again, John Levy Martin has looked at a bit in some earlier work, but the problem is he couldn't look at dependence because they didn't have the techniques to do it. And although you could just, let's just fit an ERGM to each of these networks, the problem is they're not very big. And so they're too small to actually fit models to each one. However, if you pool them and fit them jointly, you have enough power to do something. And so that's what we're going to do. So just to give you a sense of what these are like. Okay, here are the networks, right? And they're really odd. Here I've got the hue is showing the age. From red is younger, blue is older. Circles are females. The diamonds are males. And larger nodes are the commune founders. But you can see even just looking at them, they vary a lot in their density. So this is again telling us, yeah, we probably need to deal with that heterogeneity. And so we're going to use our tricks to fit a model here. I'm going to start with just the baselines. So just to show the ideas, right? If we think about forgetting all the other effects, we could have just a Bernoulli graph here, a beta Bernoulli graph where we say there's a density mixture, or the Dirichlet categoricals where we say density and reciprocity both change. What happens? Well, sure enough, as you'd expect, you get an enormous improvement by assuming that there is mixture and reciprocity. And so these things do vary a lot in how reciprocal they are and how dense they are. That's not shocking, but it's reassuring. We get what we would expect. But the fun comes when we now add other effects. So now we can start with the Dirichlet categoricals where we say, the graphs might vary in their intrinsic underlying density and reciprocity, but then we're going to control for other effects. And so here we're going to look at the gender effects, age, tenure in the organization, founders, and they take my networker card away if I didn't look at transitivity. So we're going to look at that too. So what do we find? Okay. First of all, after we take out everything else, do we see something that looks flat? No. The residual baseline acts like a Dirichlet categorical graph where the expected mutual asymmetric and null rates are about 0.818, 0.54, and 0.38. So in other words, it's a pretty asymmetric underlying process, net of everything else, as we would expect, and relatively sparse, which is, again, pretty sensible. But there are other things going on. What about power in terms of gender? Okay, we expect there's going to be a gender effect, and there is a pretty strong one. The main way it works in this particular population at that particular time is that women are less likely to be seen as exercising power over other women, and especially over other men. Men, however, don't really seem to have an effect for that. Men versus, so men are about as equally likely as being seen exercising power over other men versus women. So that's the big difference in this particular case. So that's again net of everything else. So indeed, there's a gender effect. What about institutional properties? You might expect that if you're a founder, if you're older, if you've been there longer, you're going to have more power, and that does seem to be true. So people with those properties are more likely to exercise power over those who don't have it. And then finally, what about transitivity? Indeed, net of everything else, transitive triples are favored and cyclic triples are suppressed. One thing I'd observe, though, is that cyclic triple effect is stronger. So it's not so much there's a big tendency to have transitive closure in the sense of if I beat on you and you beat on someone else, and I'm going to go beat on them. It's more that you have very few cyclic closures. So there's not a tendency that if I beat on you and you beat someone else, they're especially unlikely to beat on me. Okay. And so again, a very simple model, but you can see how we start to get insights by pooling part of the information. Does it work? Again, I'm just showing some very simple things, but indeed, if we plot all of the different some properties in all the different networks, we do a reasonable job of reproducing their overall properties. And you could elaborate it, but not a bad start. Okay, so we've now talked about what happens if we pool everything. We've talked about what happens if we pool something. Let's pool S, right? So what's in the middle here? Well, once you're in the middle of this range, right, you start thinking about general mixture models, right? That's the most natural thing to do. And in fact, that's what we're going to do. So the last case I want to show you here involves looking at finite mixtures of ERGMs. This is work that, again, I did with Fan and also Wei-Ning Shen at Irvine. And what we're doing here is thinking that, well, now I imagine I've got my graphs and I'm going to think about them. And I've got a population of graphs, each characterized by their vertex set, their covariates and whatnot. And I'm just going to think about them as arising from a set of K latent subpopulations. So I think there are K different generators, and they could be different, right? They don't have the same properties. The problem, and if I knew what the populations were, I could fit a model to each one. But the first problem is I don't know what they are. Right? So I assume there's fully latent populations. So I assume that for every latent subgroup population, there's an ERGM that generates the graphs, given the local covariates, but I don't know what they are. So the natural way to approach this problem, right, in a very typical setup, if you're familiar with these kinds of latent mixture models, so if you're an aficionado of Bayesian latent mixtures, you're like, I've seen this movie before, right? It's a very similar idea. We can describe it by this plate diagram. So we have a notion that we have our K latent groups. Each one is governed by its own parameter vector. A priori, we'll take that to be multivariate normal. There's a probability of being a member of each of those latent populations, which is governed by this parameter tau, which we take as a priori Dirichlet distributed. There's then, of course, a categorical variable that tells us which one you actually are in. And then given the group you're in, you draw the parameter that goes with your group and your covariates, and that then jointly determine your structure. And that's the typical thing. And we approach this in a fully Bayesian mode, doing a metropolis with Gibbs sampling scheme. So this is the actual algorithm. I'm sure that in the back of the room, you're all like, oh yes, I see exactly the particular thing. But the basic idea is very typical for this breed of algorithm. So, we draw one thing at a time. We draw the probabilities of group memberships. We then draw the group memberships. Then given who's in what group, we then do a pooled ERGM estimate for that. And then we go back and draw the other parts. And we just do this over and over again. And there's some technical details, but they're all pretty typical of this genre. The part that's different or interesting here is a computational challenge, and that is that this algorithm, right, requires us to use the probabilities of the graphs and some of those ratios of probabilities of graphs. And the difficulty is that probability is very hard to compute for an ERGM. It's very, very expensive to compute because of the normalizing factor that's involved. And for these things, the normalizing factor doesn't go away. So it's not so simple. You can do it with bridge sampling these days, thanks mostly to Pavel Krivitsky's work, but it's still too expensive to be practical for something like this where you have to draw it over and over again. So how do we get around the problem? Well, actually, the main method that we use is the oldest trick in the book, which is we just use the pseudo likelihood instead. And it turns out that for this application, the pseudo likelihood, which is very easy to compute, is actually quite good. It does seem to work pretty well. But you can get better results if you're willing to pay a little more for a little extra. We can sell you an improvement. We use a trick by Bornas et al., who had introduced an improved kind of adjusted pseudo likelihood. So they take the pseudo likelihood and they do basically a local sort of first order adjustment. It's more expensive, but that does improve performance. The cost is pretty high if you do it serially, but there are ways to parallelize it, and you can get the cost down to 20-30% more. So what a bargain anyway. But the bottom line is it's practical. So it does seem to work pretty well. And we can now use this to do these very general families of mixtures. So let me show you in the time I have left an example of that. And this is applied to congressional co-voting networks in the US. This is a data set by Moody and Muka, pretty famous data set, and you may have seen it before. Again, for some reason, the projector is not so great with red, blue, but you can hopefully make out the distinctions there. That's an especially unfortunate thing for a projector not to do well when you have the US Congress. Kind of a difference that we care about, you know. This is a very bipartisan projector here, I guess. But anyway. So what did they do? They went and took data on senators from Congress 40 through 115, so 1869 through 2014, a lot of US history. And they code senators based on how often they vote with each other. And they have a threshold for co-voting, which is quite high. I think it's like 75% or something in this. But so we're said to be co-voters and we vote together a very large fraction of the time. So this is a pretty strong tie. And the question is, how does that evolve? And again, the other thing is this is washed out some of my ties here. But anyway, what I've shown here are just examples of the different networks. You can see a little clearly where it's really partisan. You can see these big partisan clusters that emerge. And so what you can see even with the washed out colors is that there are real differences, right, across Congresses over time. You might think the Congress always has a typical co-voting pattern, it's just always two big groups. It's not really true. There are years like, 1869 where you might have a little cluster, but there's a lot of, little pairs that might go vote together, groups that don't really vote together with everyone, at least not all the time. There are some periods where it's 2009, not shocking, super, super tightly polarized, right? And these things vary. And why is that? Well, political scientists have a lot of ideas about that, and I'm not a political scientist, but we can observe a couple of things. One is that, of course, the composition of the Senate changes through time, and that might affect the nature of partisanship and co-voting. But of course, the social forces that govern the system change over time as well, right? And so at any given year, right, what's going on is partially a function of the mix of people there and the social forces that govern it. So it's a natural candidate for thinking about the mixture model. We can think about there being different kind of classes of senates. And if you're from a particular class, it represents a particular social milieu that generates certain behavior. And of course, we don't know what they are, but we can try to infer them. So we applied our model to that using a pretty simple model. We have mixing terms by party, and we have triadic closure, which turns out to be important as well. We observe that there are estimated to be 4 mixture components in this particular case. So there seem to be 4 different types of, if you like, regimes. And so if we do that, we can now look at the parameters within the regime. So here they are. The shading just to give you a hint about across each row what the bigger or smaller parameters are. But we can see they are really quite different. So the type 1 senates, if you like, have pretty high democratic intra-party tie rates, less inter-party tie rates, some closure, not as much, somewhat more basic. The reference category is Republican, so their propensity to tie internally is not as strong. And that's very different from, say, what happens in cluster type 4, right, where you see much stronger baseline, which is really Republican ties, real penalty to inter-party ties. But just looking at the parameters only gives us a piece of the picture. One of the things that's great about being able to simulate, right, is that we can do what ifs. So we can go back and say, let's just take the entire history of the Senate and then say, what if all senates had been type 1? What would the world look like? What would if they had all been type 2? What if they'd all been type 3? We can do these things, right? We're computers. It's awesome. And so there's a lot of things you can look at. What I'm just going to show is one thing, and that is intra and inter-party triangles. So why triangles? Because these are minimal voting coalitions, right? You get 3 politicians that vote together all the time, right? So that's a building block of other coalition formation, which is important in political matters. And so here I'm showing the inter-party triangles, red and blue. So blue here, red here. That's for Democrats and Republicans. These are the inter-party triangles. Scales are different. You always tend to have more intra than inter-party as you'd expect, but they nevertheless do vary. So we can see if the world were like a type 1 Congress all the time, what would we see? We'd have a lot of inter-party cohesion for the Democrats, not much for the Republicans, and not much cross-party ties. So this is the Democrats are all over here, and the Republicans are okay, only different. But if there were type 2s, there's not a lot of coalition formation in general. Everyone's kind of all over the place. Maybe this is the ideal world for some people, but it's not the world we're in all the time. But there is a time when we're in that kind of world. there's a type 3 world. Maybe this is more like our kumbaya world, where there's some intra-party cohesion, but there's more inter-party in relative terms cohesion, although again, there's a limit to how much there ever is. But you see more of it on average there. And then finally, cluster 4, there is actually more inter-party than there is sometimes, but the intra-party is off scale. So here you have really strong polarization on average. So as a fraction of all coalitions, this is much smaller. You tend to have really strong local polarized solutions. So, we find there are these different 4 things out there. We don't go beyond that to say what it all means. That's what political scientists do. But what it does do is it gives an illustration of how we can use these kinds of techniques as a probe to start asking those questions. And in fact, I don't think this is the probe to answer the questions at the end. This is a way to start asking them, and then you'd want to do a lot more analyses to probe what's going on. But now you start to know where to look, right? You can classify years, and you can, what's different about them. Now you can ask why. Without that, you just wouldn't know where to begin. All right. So I want to wrap it up to make sure there's time for questions and whatnot. So I will stop here. So hopefully, if nothing else, and you have no idea what I've been talking about, which is possible, and that's okay, don't tell me. I hope I have convinced you that these kinds of multiple network problems are really a rich area, both for applications and for method development. There are a lot of cool parameterization problems. There are cool computational problems, statistical problems that give us neat things to do and write papers about, which is fun if you're a methodologist. There are a lot of strategies to this. And so one thing I want to emphasize in the end is that there's a reason that there are multiple different solutions presented here. Right? There is a certain philosophy that's out there. Maybe some of you have it, but it says, we should just deal with all of this stuff by making the world's most non-parametric model. Probably not even a statistical. This has parameters in it. We could put a neural net on this thing, right? And we could just pour data into it, like we pour things, a trash can into a dumpster, and then we could just run it, and truth would pop out. It would solve our problem. We don't have to think ever again. Okay. You might suspect I'm not sympathetic to that point of view. The world's a complicated place, right? The problem with there are many problems, but one problem with, why didn't we just use the method we use at the end for everything? We could just do everything the super non-parametric way. Well, it's consumptive of data. For one thing, it's expensive, but it also, right, by not imposing structure that should be there, right, we limit our ability to reason what's going on, right? So the most non-parametric solution is not always the best one if we have questions we can answer. For instance, we started out with a question where we know there's a unifying generative process. We know there was because we made it, okay? And we want to understand and approximate it. In that context, simple pooling is obviously the right thing to do. In other cases, we may be doing a very exploratory analysis. We want the flexibility. So the key is we want to know, what do you want to do? What do you want to capture in your problem? What do you need to prioritize? Do you care more about data efficiency? Do you care more about computational efficiency? Do you care about being able to implement the method? Do you care about understanding the result? What is it that you need to do? And there's going to be room for a lot of different schemes to address the different needs. So there's never going to be a one-size-fits-all thing. So if nothing else, take away the fact there's more than one solution, and they're useful for different things. And I think it's important to keep that in mind, because there are some people out there who really want everything to be one-size-fits-all totalizing. It doesn't work. Okay. Second thing, this is more aimed at the children among us in relative terms. Okay. Which I am no longer myself. You want to have an impact on what people do. You want to have methods that get used. And maybe the answer is no. So, if you do, you want your tools to be things that you can implement using standard techniques. You want to be things that you can run without a supercomputer. You want to have things that don't require giant mountains of data, right? Again, not everyone's, but we're in an era where there's a certain more is more philosophy. Right? It's all about get the biggest data, the deepest learning, right? And pour it into the most expensive computational solution that you have to be a Google engineer and run it for a week, on all their machines to get an answer with the world's fanciest gradient descent. You can do that, but random people out there in the world don't have those resources. They're not going to be able to use the tool. So if you can come up with ways to do what you need to do in ways that can plug into existing techniques, leverage existing formulas that people are familiar with, that you can work with existing computation devices or whatever, your stuff is more likely to get used and you're more likely to have an impact on practice. So that's just a shout out to, think about when you're thinking about the sets of tools and things to work on out there. And I know this is a crowd that cares about implementation and cares about software and cares about, which is not true of most crowds, okay, that I talk to. So it's something to think about, right? The most macho solution makes you look cool to your friends, but it may not be the best way to go. So it's just something to keep in mind. Right, so at the end of the day, goals matter, right? You're not going to get everything right. No method's going to do everything. But if you look for models that are adequate for purpose, you can often do quite a lot. All right, so with that, let me close again by recognizing my group and my collaborators on the Tobias lab. Doug, Alfredo, the students Eric and Vera worked on some of the simulations. Of course, and my group, Liz worked some on the protein work, and of course, Fan also worked on statistical stuff. Some of these people, fine folks, may be available for postdoc hires, so something to keep in mind. So I will wrap it up there. Thanks to all of you for your attention, and I'm happy to take questions.
Could this framework be extended to like ERGM or WRGM type models, do you think?
Yep. I mean, why not, right? If you can do it with one graph, you can do it with a graph series, and you can think about sets of graph series. So sure, you could use, of course, there are different, I talked about 3 different methods, but yeah, in general, these kinds of approaches can be used with ERGMs or multivariate ERGMs or whatever. It's just everything's going to get more complicated as you add more parts. So you have to think about, is that what you want to do? But yeah, there's no particular reason why you couldn't generalize these techniques.
I had a question about making your methods available to practitioners, especially with the last one where you talked about political polarization voting blocks, because a lot of times talking with political scientists, they're like, oh yes, we want to use the newest fanciest techniques because if you do ML in a political science paper, you're going to get published. But also there was that paper recently from Princeton, Arvind, that was saying a lot of social scientists are using ML models kind of wrong. So how do you deal with the tension of you want these models to be accurate and correct, but also simple enough to be usable correctly by people who aren't in the field?
Yeah, that's a great question. And that's a great question because, you could talk forever and everything you say is defensible to somebody or other. So, do I have the right answer? No, but, I'll tell you something. I think let me start by observation / anecdote. So if I think about one of the people who's been most effective in my personal judgment at this type of problem over the years has been, of course, now I say the person, I'm going to immediately blank on his name, Adrian Raftery. And Adrian is, famous for a lot of things and has made a lot of important contributions. But one of the things that has made Adrian really, really famous is that he's done a lot of work to try to come up with things that people can use who are not statisticians that improve what they do. And I remember a conversation I had with him some years ago, and he mentioned some of the fights he'd gotten into about the BIC, right? He didn't invent the BIC, but he wound up popularizing the BIC among social scientists. A lot of them know about it through him. And he got a lot of pushback because, and if you're a statistician, there's some in the room, why? Well, the BIC has all these limitations, blah, blah, imperfect, blah, 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 right? Okay. And all that's true. And he's a sophisticated guy. He knows that. He's like, so I know the BIC is not magic, it's not the end all be all. I know that. But what I thought about is, okay, what is something I can give a typical social science person fitting data that they can use that will be better than what they do now? And at that time, I think a lot of what people were doing is just doing model selection by null hypothesis tests, which is not a great way to do model selection in general. And he's like, look, this is better than that. And he's like, if I can get them to the point where they can have a conversation about why this is imperfect, then I win, But I want to figure out how do I get them from here in the ditch? That's my language, not his. But I own it because, man, I lived in that ditch. It's a ditch. How can I get them out of the ditch, right? How can I advance them? And so that's one philosophy, is to say, okay, can I look around at what people are doing and say, can I give you a method that's a little better than what you do now, but still easy? Now, I mean, there are other points of view. Other people say, well, you should just never give people anything that's not perfect, and that way they won't screw up. There's another mode that says you should give people lots of rules, and that is a kind of an ascendant view. So what we can fix all our problems by having lots of rules that you have to follow for every analysis. And this is a kind of, in my mind, a cargo cult model of statistics and science, right? If we build the airstrip and all this stuff, the planes will come and land and bring us the cargo. Okay, if we go through all these motions, right, we'll never have a replication problem. Yeah, right. The problems come from not from the tools, but from not thinking if you don't engage your brain. Right? Psychologists have had problems because they ask stupid questions. And then they got P less than 0.05 and said, well, it must be true. Well, the problem wasn't the poor p-value. I mean, the p-value is just a yardstick. You're like, okay, is this effect, here's what, here's what I would expect the variation. Is your effect bigger than the yardstick? No, yes or no. That's really all it does, which is useful. That's a pretty helpful thing to do. But like, that's not a magic determinant of truth. So, if you're a null hypothesis, if your alternative hypothesis is that, I'm a flying unicorn. Oh, like, well, p less than 0.05, a Bayesian would say, yeah, no, that's not it. But if they engage their brains, wouldn't have had that problem. So, I mean, rules, having rules, right? Well, we'll just create a rule for that isn't going to solve the problem that you have to actually think and use your substantive knowledge. The problem is not a statistical one. It's failure to use substantive knowledge and think about if people are having hypotheses, like if I'm outside a box, I'll be more creative because I'm thinking outside the box. You should probably think, okay, that's not a good thing to do. So I'm just my very opinionated take, obviously. But I mean, I think there's no one answer, but I think a multi-pronged strategy can be helpful. One, try to come up with methods that are relatively usable and intelligible to the population you've got. Recognize that you have more than one population. You're going to have some sophisticated users. You're going to have some not so sophisticated users. Talk to who will listen to you and make the improvements where you can make them. Try to give people reasonable ideas about how to use things, but warn people they're going to have to think. And then at the end of the day, I think to improve the quality of work, we have to teach people. So we also have to try to train them to get the quality of expertise up, which is, there's a limit to that, how that can be done, but it can be done. People can become more sophisticated. So I don't think there's a magical solution to this problem. And then, some fields are notoriously bad about people develop these ideas that, well, you can only use these models or those model. And all you can do with that is, call people names and, tell them they're wrong, and maybe they listen to you, maybe they don't. But if you at the end of the day, if you do good science and you get good results, I have this probably unreasoning faith in the sort of long run self-correcting nature of science. That results tend to work in terms of getting people to follow you. If you have real breakthroughs and they're really clear, then over time, people are going to emulate what you do because it works. And if people can have all kinds of rules, but if they don't generate real results and they're not predictive, they're not useful, in the long run, people will get tired of their game. So I wish I could give you a better answer, but that's my answer for fighting the good fight. But I think all of us who are interested in trying to improve methodology, what we can do is get out there, teach people, try to make stuff, try to put things in people's hands, try to come up with practical solutions we can, and hopefully by the combination of all those things, we'll make the science better. Fingers crossed. Mm-hmm. Yeah, David.
When you had the slide up there towards the end there about being good for practitioners, I thought at first what you meant was practitioners, like
who's a practitioner is relative.
What are you talking about as a practitioner?
That's a great question. Right? Yeah, it's all relative, right? So and I meant it in a relative way. Right. So if you're in the esoteric end, your practitioners are probably the power users of the community. If you're kind of a power user and you're making developments, that your practitioners are maybe working scientists who are not that methodologically sophisticated. If you're a random working scientist, your practitioners might be people outside the scientific community doing stuff. I mean, so it varies. I mean, I work on, I'm an esoteric person, of course, by all accounts. But every now and then I do work on things that, that people out there in the rest of the world have to deal with, meteorologists and broadcast meteorologists. There's people trying to figure out how do we inform the public about disasters and things like that. And obviously, I'm not saying we'll go fit an ERGM, and that's not what we're doing. For them, we're trying to get answers that are evidence-based that we can distill down to simpler heuristics. So that, we're talking about practitioners at that level, of course, it's far more basic than what's on this slide. But I think the general principle of what I'm suggesting applies at every level. That is, you take whatever you're looking at and figure out, okay, how can I package the pieces of that in some form that my audience can use and that interfaces what they have. And so here I'm mostly talking about, okay, multi-network ERGMs. So this is pretty fancy stuff. So I'm talking mostly to a room of pretty fancy people, even if this isn't what you do. This is a fancy group in general, but everyone here who's making stuff is packaging something for somebody in some way. And I think, if you're making a tool of any kind, thinking about how can I make that tool interface with people and my user base it's relevant.
And
So I intend it as a general idea, although I can also give a more narrow argument for this specific work, but it's probably not.
so then the follow-up to that is, do you have these alternatives implemented now in StatNet such that they would be available to us practitioners?
Well, the beauty is you can, at least with the very first thing I talk about, you can download code with the paper, but the beauty of that is that you don't have to modify anything in StatNet. You can do it using the existing code base. You have to, there's some very simple pre-processing you have to do. But yeah, the code is with the paper. I do hope that the code will be in StatNet eventually. That's not literally packaged in StatNet, but you can download it and run it. And it's, again, you don't have to modify the tools at all, which is great. The second one will be in StatNet, but it's not there yet. But the key is we can implement that and stick it in. The third one is pretty esoteric, and yeah, you can download the code with the paper, but that one is not yet. So, that's not very user-friendly at the moment, unfortunately. So again, I didn't have the thing about how this is nice and user-friendly with that one. That's the one that's not very friendly, but it's Mas Macho. So, that's the trade-off.
Yeah, I think it's really cool to see that, because in my limited view, I consider ERGM a very social science, like social network method. And it's cool that it fits physical problem really well. So my question is, like, when you think about network method mostly for social science, how do you know when it would apply well to other problems? And conversely, if you're someone developing method, what field do you keep in mind?
Yeah, that's a lot of fun. I mean, that's something that's one of my long-term interests, is figuring out how we can take ideas from one part of science and use them in other parts of science. And that's something I've been interested in actually since I was undergrad. So in some sense I trained to do it. So one answer is, okay, go back to your undergrad and take a whole bunch of physics and other stuff. But part of it is you have to be alert to where there are symmetries between the assumptions of the model you use and the formalisms of the model you use, and the formalisms and assumptions that are relevant for some other problem that you encounter. So in the case of the, and I don't know if anyone wants to see it, but maybe since people are narrowing down, I'll show you really quickly. We've got some part of the trick for this one case, just to illustrate the idea. Right, so here's an ERGM, right? This is how we think statistically about ERGMs, right? This is what we put up there, right? And we have these parts in the model, right? Graph structure, parameters, or reference measure, all that stuff. Okay, but if you do StatMech, right? Here's how you might think about the world, at least in equilibrium. Okay, you've got a model that looks like this. And so you've got this thing that's a multiplicity. You've got, this Hamiltonian adjusted by Boltzmann's constant times temperature. You've got your partition function and so forth. And the beauty is to realize that I can rewrite this in ERGM form, or I can rewrite the ERGM form as this. So I can re-express the ERGM in terms of an effective Hamiltonian. That's an energy function. And so here, part of the trick is that the math turns out to be exactly the same. And it's exactly the same. It's not just similar. It's precisely the same. And there's actually deep reasons for why these things are connected. The exponential families, if you go read James's work, you can see there's a connection between exponential families and statistics and the physical interpretation. And he makes a lot of claims about it. Not all of James' claims are true, so we have to be careful about James. But the basics do carry through, at least for this sort of thing. And that's the beginning. That's not the end of realizing that you can use it, but you start realizing, hey, there are these similarities. Now, can I exploit them in a rigorous way? And to do that, we do have to do some complicating things. You have to keep in mind every system is different. So there are things, for instance, we want to model, amyloids. So here's are just for fun. I'll show you the networks that come out in amyloid. They're very different. So these are aggregates. I showed you unstructured aggregates. These are aggregates that form in Alzheimer's disease, in Lewy body dementia, and type 2 diabetes, a whole host of diseases where proteins will aggregate into these insoluble linear structures that have these really interesting forms. These are, by the way, all of the topologies that have been found today. We went through the protein data bank and we worked out all of the fibril types, and there are only 5 that have been seen so far. Everything falls in these categories. It's the frequencies. And so they have these really interesting kind of linear forms. And then the question is, can we recapitulate that? One thing that's very interesting, by the way, is these things, the system converges into these things, it stays there. So these are degenerate structures, right? So you've heard of degeneracy, it's normally seen as a bad thing. This is the real world being degenerate. And the question is, can we model that in a reasonable way? And we can use ERGMs to do that, but we have to correct for things like bond energies, and there's a lot of stuff we have to think about. But there are ways to do that rigorously, and it's a longer conversation how to do that. But in essence, the first piece of how you get started is recognizing, hey, there's a similarity. And including, we know how to make ERGMs that congeal into weird forms. And usually we try not to do that. That's considered a problem. It's bad. But here, this is what actually happened. Now, it is bad when it happens to your brain. It's not good. But we know how to do that. And what's cool is we can. And now I'm going to show you. I have an excuse. Thank you for giving me this. I'm going to show you something. I got to get to the end of it. Oh, here are the. So this is our, we can simulate. So we're using ERGMs. We can simulate all of these different fiber forms. So we can recapitulate all the forms that have been found so far. And we can look at the phase structure and all that. But we can also do kinetic versions. So we can add dynamics to the ERGM. Now we have to add some assumptions. We do this by just assuming basically local Arrhenius-like kinetics. And it turned out this is also fun because although we independently re-derived that, we then realized belatedly that thing we had derived was exactly the same as a particular dynamic model family called the ERGMs, or longitudinal ERGMs, that Johan Koskinen and colleagues developed earlier. So in fact, we accidentally rediscovered the ERGMs. But that was neat because theirs was a completely social thing. They had no physical interpretation in mind. And we had a completely physical justification. It was motivated by chemistry. And it turns out they give the same formalism. Okay, so we can then look at the kinetics of fibril formation. And I'm going to show you a movie. I'm going to show you a fibril formation. This is fun. Okay, this is fun. This is so cool. I never get tired of this thing. And when this thing popped out of my simulations, it was amazing. All right, so you may not be as excited. I shouldn't talk about it. I'm excited by it. So this is a formation. We're going to see the formation of something called a 2-ribbon fibril. So it looks like a ladder. This is a real thing. These things are really found. We're going to start with, in the model, we're going to start with a bunch of monomers. These are going to be, it's an empty graph. That is, you got a bunch of proteins that don't hook to anything else. So imagine you have them in a little cuvette or something, a little aliquot of sample, and they're going to be bumping into each other, making ties. We don't simulate the bumping into, that's all tacit, although we have to account for it. Again, there's interesting things about how we do that mathematically. But we're going to think about a graph process where proteins are going to hook together. And so let's see how that unfolds. So at first, right, here you've got your monomers. They start bumping into each other and they start aggregating. And time, by the way, is moving faster as we go through the simulation because the real process slows down and we don't want to be here all day. So the first thing it does is it starts to aggregate. You get formation of little tiny oligomers. They're kind of boring little dendritic structures. And they grow larger and larger. And it looks a lot like a typical random graph formulation. If you've seen them before. And it collapses into this giant component that is pretty unstructured. And in fact, it looks like a big blob, right? So you think we're done. Nothing interesting has happened. Nothing will occur. But we're going to let it keep running. And again, we're not driving it. The thing is just evolving just naturally through time. And if you look at this blob, so this is like a gelatin, if you think of what it physically would be like, it's like a gel phase, which we do see experimentally. You see there are a few of these little nodes that are yellow. So notice yellow if it is locally in a 2-ribbon conformation, okay, and mixed in with this blob. You see these yellow dots. And if you look, you see we're getting a few more yellow dots. So this thing is just kind of randomly evolving. You think nothing is going on, but there are more of these dots. And if you keep watching it, something interesting starts to happen, which is that blob starts to change. It starts to sort of pulse and it starts to self-organize. And if you look at what's happening, you start seeing that there are more and more of these yellow pieces. And the yellow pieces hook to each other. And if you keep watching it, something really astounding happens. And all those little fibril pieces start to self-assemble. And the entire thing unfolds into this beautiful two ribbon. And it does it entirely spontaneously. There's spontaneous self-assembly. That's a 4-parameter ERGM. I can do that with 4 parameters. That's really quite profound. So we can take this whole process on a laptop, we can simulate this thing. You wouldn't be able to do it in MD with a 4-parameter model, and we can recapitulate that whole process. And along the way, right, it goes through these interesting phases. This tells us there's a lot we understand. Prior to this, I don't think, maybe somebody knew it, I didn't. I don't think anyone knew that we could get those kinds of well-ordered structures by going through an unstructured intermediate phase. So in fact, while I was simulating this and trying to figure out, I had an idea I could make these ribbons. I thought that what would happen is you get little pieces of ribbon and then they would assemble. And in fact, a lot of people think that that's what happens, or thought that was what happens. That's not what happens, right? It first collapses. And then you start getting these little intermediate structures. Those things self-catalyze the formation of more little immediate structures. And then they feed on the blob in essence and convert it all into this two ribbon. And so this is even a simple model as that. And it has really simple things in it, like NSP2s. There's nothing exotic in this model. It has really complicated behavior. It can assemble and it goes through these weird phases. We don't understand this stuff at all. There are really interesting, deep questions to be asked. But what's cool is, again, that process, and we think, we didn't plan for this thing to have this gel phase in it. But if you talk to people who make this stuff in the lab, they're like, yeah, it goes through a gel phase. And what's also fun, we had to fight with reviewers because a lot of reviewers didn't think it goes through the gel. Well, this isn't realistic because it has this gel phase. And we had to dig out these old papers that showed, no, it goes through the gel phase. Because now what they do is they get the final fibrils at the end of the experiment. They clean them all up. They put them like in an grid or something. And so they only have these beautiful fibrils. And then they solve for the idealized structure. That's why you see things like what I showed you with these idealized fibril forms by NMR or sometimes crystallography. But all that intermediate stuff is lost. Another thing that we see that is, again, there's experimental evidence for, it's not well studied, are defects. So along the way, you get these little failures in the fibril, right? And that makes sense. No real world thing is perfect. People usually ignore these defects. Those defects, though, are potentially a way we could fit the model, right? These are observables, or potentially experimentally observable. I talked to one group, and they were talking about some drug candidate for Alzheimer's that was failing, and they think that what's going on is that the drug develops an affinity, basically just sticks to these defects. And, so potentially we could detect the defects by developing these that bind selectively to defects. And then since different kinds of mixtures may make different ranges of defects, that gives us a clue as to what the model parameters are. So I think there's just a huge amount that we can do in this space. It's just almost entirely untapped. So, I mean, to come back to your original question, how did I get there? It's a long road and it's complicated. And I can't tell you how you'll get there. But if you stay alert for something where you have a trick, and you know how to solve certain kinds of problems. And when you see in the world, you look at what people are doing, you realize, hey, my trick could be applicable to that problem that person has. Maybe I can be useful. Then you can do it. And then you've got to find yourself good native guides. You've got to have people who are domain experts who know about this. I collaborate with people who are experimentalists who do this stuff for a living and know the stuff and can tell me when I'm full of crap, which is often, And then you have to, you do that and you subject yourself to peer review in that area. We don't publish this in the social network community, right? We publish this stuff in the journal Physical Chemistry and other biochemistry, places like that, where we're getting reviewed by people who are in the field, which means sometimes we have to deal with dumb objections, but we have people who know something evaluating. And if you can go through all that and you can get it through, maybe something you do will be useful. And that's, of course, what we all hope to do. So anyway, there's your answer.
Super. Well, this has been amazing. I love that last video. We should probably move into more individual conversations now, but let's thank Carter again.
Thank you.